Oh, hello, welcome. You're at Jimby Live for our September 29th, 2023 edition. We're very delighted to have Sharleen Flowers and Stephanie Gardner here to talk about their paper, Here is the Biology, Now What is the Mechanism? Investigating Biology Undergraduates Mechanistic Reasoning within the context of biofilm development. I'm going to put the link to the open access paper into the chat shortly. During our webinar today, attendees are in listen and view only mode. And when you have questions, of which we do encourage you lots of questions, please add them to the Q&A box, which you can find Q&A in your Zoom lower toolbar. They're going to give a talk, an overview of their paper, and we'll have lots and lots of time for your questions at the end. So welcome to our authors, and I'll see you after your presentation.
Okay. Good afternoon, everyone, or good morning if you're on the West Coast. We're really excited to talk to you about our research. I'm going to kick us off. So I'm Stephanie Gardner at Purdue University, and I'll hand it over to Sharleen Flowers, who's at the University of Colorado in Boulder. And so just to give the broad context for the education research project that we conducted, it's really grounded in modern day biology and the drive to understand and describe complex systems. And so if we look at this figure over here, you can see that single species and multiple species exist within highly networked, connected communities. But really to understand the functioning of those communities, we need to look down across many levels of organization, and that's just on the physical scale, in addition to what's happening over temporal scales as well. And so in this example, right, to understand multi-species communities, one needs to understand things at the individual level, the cellular level, and the molecular level to fully describe and understand those. So within the context of biology education and the vision of change, that students need to develop competencies in systems thinking. So examining the system as a whole and its parts, and then thinking across levels of organization and recognizing dynamic interactions. So this is an example figure from a paper from Duncan and Reiser, really looking at this interplay and going across levels of spatial organization or physical organization to understand the function of the heart, needing to understand tissues, cells, proteins, molecules, gene regulation, for example. And this is critical for explaining biological phenomena. But work in education research within biology and other disciplines has revealed that systems thinking is complicated and difficult. So for our work, we drew on a couple of different theories that nicely lined up with our disciplinary context, our biological context, to help us understand and characterize students' thinking. And so we used two frameworks. And so we wanted to briefly give you an overview of what those were. So the first framework that we used was the theory of knowledge integration. And so this theory is in the context of students learning in the sciences, is that students have pieces of knowledge that they connect in a variety of different ways to form knowledge networks. So they process new information and organize it into their existing knowledge framework to make sense of biological phenomenon. And so this was used in a paper examining students' knowledge of DNA replication and transcription and translation, for example. So in this case, they use this framework to characterize student knowledge. So in this little bubble here, it's looking at what they saw as fragmented knowledge, where students thinking about what entities, what things are involved in DNA replication, you might recognize that there are some relevant entities, but they're not well connected. So they characterize this as students' knowledge being fragmented. And then they identified transitional student knowledge networks, where they're starting to make connections between relevant processes and entities and actually bringing in different processes. So in addition to DNA replication, thinking about, transcription, for example, and then in their data set, what they saw also as connected was now contextualizing these processes and the players or entities involved into broader processes within cells, so cell division and protein synthesis and translation, for example. So they characterize in their data set, Southard and colleagues, drawing from Clark and Lynn's framework of knowledge integration, a continuum of student knowledge in this particular context, from fragmentation to integration. But as you know, right, it's not just connecting these, it's more than just saying things are related to each other or lead to one another. Another framework that is linked to biology is thinking about mechanisms. So, right, so drawing connections and describing that target phenomenon and identifying the components involved and how they interact over space and time to produce the phenomenon of interest. So, for example, the DNA replication, right? There's more to it there, and lots of proteins and DNA itself, and lots of processes going on that make up the mechanism of DNA replication. So they're composed, like I said, of mechanisms are composed of the things or the entities, their activities, the way they interact with each other, how they change their function, and the conditions of space and time. And so mechanistic reasoning is that approach to understanding and explaining and problem solving around biological phenomena. So those two frameworks are useful in our particular context, which we'll tell you more about.
Thank you, Stephanie. So we wanted to investigate this in a particular context, and my disciplinary background is in microbiology. And so this research was part of my dissertation work. So I wanted to put a little bit of flavor into this, a part of myself. And so biofilms was really attractive for me. As we know, biofilms are very significant to science and society. So if you're unfamiliar with what a biofilm is, it's a complex system in which individual bacteria aggregate together on a surface and build a sticky matrix, which we can see that formation occurring in the figure on the right, where these bacteria are adhering to a surface, and then they start to divide. Other bacteria come in. They are emitting these proteins that help build this structure that eventually kind of blossoms into this mushroom shape. And eventually at the end of biofilm, if it gets too big, it then disseminates. So biofilms are a very important process because they are ubiquitous in nature. It was previously thought that bacteria dwell in they dwell as individuals, but we've now come to find out that bacteria actually dwell in these really interesting biofilm formations. And so these have significant impacts for health and the environment and infrastructure. For example, biofilms can build in catheters and on pacemakers and can cause really serious bacterial infections that are resistant to antibiotic treatment. And so ASM has recognized how important and significant biofilms are, such that the American Society for Microbiology curriculum guidelines recommends the instruction of biofilm formation as part of microbiology curriculum. Yet despite their importance, there are very few empirical studies conducted on student learning of biofilms. And so we recognize that there is this need to fill this gap. And so that's where our study comes into play. So the rationale, as I just described, is that there's this gap in the literature describing how biology students use mechanistic reasoning within systems to understand and reason with biofilms. And addressing this critical need will provide the necessary foundations on which to build future research-informed teaching approaches for university educators. So we sought to answer the research question, what are the features of biology undergraduate students' explanations in the context of biofilm development? In order to answer our research questions, we recruited 9 undergraduate students of various class standings and biology majors at a large public Midwestern university with very high research activity. We held semi-structured think aloud interviews with our participants, and I'll briefly describe what that means. Semi-structured meaning we have an interview protocol that the interviewer asks a series of predetermined questions, but it's semi-structured in that there's flexibility to add probing questions or clarifying questions. And then the think aloud, what that means is the interviewer is asking the participants to narrate their thoughts as they reason and think through the tasks. So these interviews were conducted in 2019, and it has the structure of the interview is seen at the bottom of the slide. So at the beginning of the interview, the students defined and related 3 biological subsystems, which we have defined as gene regulation, cell-cell communication, and phenotypic expression. We chose these 3 subsystems since they are relevant to biofilm formation and are also very important in understanding biology in general. So after the students discussed the 3 subsystems, we then moved into the biofilm context. So this started with orienting students to a biofilm figure, which we will show shortly. And that figure was to help give the students some basic information about biofilms so that they could engage in the interview. After discussing the figure, the interviewer then asks them to explain how a particular transition point occurs, which we will see in that figure. We use their verbal descriptions of the explanation of that transition point as the basis for our analysis. Specifically, we used thematic coding to characterize and analyze their responses to help us form a pictorial representation of their response. And as Stephanie mentioned earlier, we're investigating their knowledge networks. So we use the theory of knowledge integration to help guide the characterization of their knowledge networks. And 2 researchers then discuss these characterizations to agreement. So here's that figure I mentioned earlier. This was provided to students to help orient them to the biofilms context in case they were unfamiliar with biofilms. This figure came from a scientific review and synthesis of various relevant biofilm literature to create this model, and it was also vetted by 2 microbiologists. So see with the red box that's about to pop up, this is the specific transition point that we asked students to describe. So initial attachment to irreversible attachment. Okay, so let's dig into the data. So we found 4 main ways that students explained this transition point. You can see the 4 titles, so no links, linear linkages to phenotypes, subsystems converge on phenotype, and interconnected phenotype. You can also see the N value, how many students describes their explanation fits into that category. And so these, what we're seeing here are various exemplars from each category. All the students, except for 1, described links between the subsystems when explaining the transition points. And as we can see here, the most common links were between cell-cell communication and phenotype to phenotype, as Stephanie is showing in the upper right, the linear linkages to phenotype. These connections were very common amongst our students. We also saw that the majority of the mechanistic models had at least 3 subsystems causally linked in one chain of connected ideas. Again, the linear linkages to phenotypes is a great example here, where they have 4 different subsystems that they have connected in a causal chain. So I can go over briefly what each of these categories mean. So the no links, we're not seeing any links between the subsystems. The linear linkages to phenotype, we're starting with some sort of subsystem. And then there's a linear description to phenotype, then to other phenotypes. The third one, subsystems converge on phenotype, seeing similar ideas of two subsystems being connected. But what's different and interesting about this one is that there's two subsystems that are converging and have effects towards one, as we're seeing kind of at the bottom. Yeah. And then the last one, interconnected phenotype. We're seeing that there's all sorts of different subsystems that are being described and connected. And what's really interesting is this idea that the phenotype is part of this process, is being interconnected and has effects on other subsystems. So that one arrow that Stephanie's pointing out, where we see that the cell aggregation, that phenotype then has downstream effects towards the subsystem of cell-cell communication. So we compared these student models to a normative model of biofilm development. You can check out that normative model in figure 2. It's quite large. But we based that model on information gathered from our literature review. And when we compared our normative model with the student models, we found that most of the students lacked the correct connections. In particular, they were describing these phenotype to phenotype or cell-cell communication to phenotype connections. And we rarely saw any cell-cell communication to gene regulation connections and gene regulation to phenotype connections that we would expect to be part of this transition point. So these were rare in our data. And this was because a lot of students did not include cell-cell communication and gene regulation as subsystems in their explanation. So we saw these models, and we wanted to characterize those arrows in between the different subsystems. So how are they describing that linkage between the subsystems? And so we, informed by previous work and from what we saw in our data, we came up with 4 connection codes to describe these connections. So mechanistic, which is what Stephanie described earlier, where we have this causal sequence of events that includes lots of entities and activities. It goes down a level of organization, as Stephanie talked about earlier. Being able to transition across levels of organization is very important in describing this mechanism in particular. Specified causal, we're having some causal factor that's being described as part of the linkage. Unspecified causal, there's a linkage that's described, but no causal factors are included. And then the associative connection, where it's a very unspecified link and no entities and activities are described. So we took the 26 connections that were described by our 9 students and categorized them using these connection codes. Well, if Stephanie, if you could go one, perfect. So we saw that associative connections, we were very pleased to see that this was lacking in our data set. But we were also, I guess it wasn't a surprise, but we saw that only around 15% of these connections were actually mechanistic. The bulk of the connections was this unspecified causal. So it's good that there is some causality, but the unspecified part is we would like to see more specified causal or mechanistic links between their subsystems. We then investigated the various features of their student explanations. We were looking to see, did they include all the correct knowledge that one would need in order to describe this transition point? So in that first column with number of entities, we saw that no students included all 9 expected entities. The most missed were talking about extracellular signals, receptors or signaling cascade, and different transcription factors or proteins that are involved in DNA or gene regulation, as well as genes. We also saw in the next category that only 2/3 of our students discussed all 3 phenotypes. So the 3 phenotypes that we saw between in that transition point, these cells aggregate together, they lose their flagellum, and then they produce that sticky matrix. So we were looking to see how are students describing how all 3 of these phenotypes are forming. Only 2/3 described all three. And then in this last column, we were looking to see, are they describing the two subsystems of cell-cell communication and gene regulation? The third subsystem of phenotype is represented in that middle column. We saw that only 2 students out of the 9 integrated both cell-cell communication and gene regulation into their explanations. 6 out of 9 students included cell-cell communication, but only 3 out of the 9 included gene regulation. Both of these subsystems are incredibly important in biofilm formation. We then investigated the level of organization and localization of the features of their explanations. So the level of organizations, we looked to see what sort of things were being described, and then the level of organization of that thing. So for example, if they describe genes, then we would give them a black box for macromolecular, since they've described a thing from that level of organization. Go ahead, Stephanie. So we saw that cellular and macromolecular were highly enriched in from our students, which is great. We saw that these undefined levels of organization, so they were describing either something unspecific or something that is larger than these levels of organization. So a little bit of undefined things in their explanations, and some environmental. And then not many molecular entities were described, which as we'll see later, being able to describe things across these levels of organization are important in describing the transition points. So missing out on that molecular level has some consequences. Then localization refers to where are these various processes that the students are describing are localized to. Since we were looking at these bacterial cells, these localizations are contextualized to the bacterial cell. So go ahead, Stephanie. As we see, the bulk of students describing these processes are either unspecified, processes that are occurring outside of the cell, or at the cellular membrane. But unfortunately, we did not see many processes being described inside the cell. It's excellent to see that they're describing things that are going outside, happening at the membrane. But a lot of these, the gene regulation process, for example, that's happening inside the cell. And so we're seeing that students are missing out on important processes that are going on inside the cell that ultimately lead to the phenotype that's observed. So using all of this data, we then made a model of student knowledge integration within the context of biofilms. And so as we can pop up the first one. So this is similar to what Stephanie was showing earlier with our fragmented knowledge. So as you'll see, the text and arrows in these models represent characteristics that were very common. So the black arrow, we saw that a lot, the text that's black. Whereas the text and arrows that are in the darker green represent subsystems and connections that were less frequent in that category. And then text in the very light gray, or if there is not an arrow present, that was not characteristic of that model. And you'll see that the opacity of the images also is similarly aligned to how we drew this. So we're seeing with our fragmented models, these contain the fewest amount of connections, mostly between the phenotypes. Some students' fragmented models contain that connection between the cell-cell communication in the bacterial phenotype, but none of the fragmenting models involved gene regulation. So these fragmented models are the least specified, and they are the least correct. For our transition models, these incorporate more connections, as we're seeing with the increase in arrows, that are including more cell-cell communication and gene regulation subsystems and their effects. The content and the nature of the information described in the models is more specified and more correct than what we saw in the fragmented, but we're still missing some key characteristics, such as the connections between cell-cell communication and gene regulation. And then our last model is connected. So these have the highest amount of connections, and they are the most specified and the most correct compared to the other two models. You see that all three of the bacterial phenotypes are being explained using the subsystems of gene regulation and cell-cell communication. And the correctness and the nature of their content in these models are the most correct. So we have our N values shown here at the bottom. You see the majority of our students are either in fragmented or transitional knowledge, with only one student that we characterize as connected. So we see that there's still some growth that can be had in this area. So to conclude this study, we saw that many students in our sample may not possess an integrated perspective of biological systems, as we saw on the previous slide. We saw that there were isolated ideas with few connections, and that they were missing some important entities and subsystems to help with their explanation. So, for example, the transcription factors and genes were often missing, and that's pretty consistent with students, with the literature that shows that students are uncertain about the role of proteins and genes. We also saw that gene regulation, related to what I just said, is a possible sticking point in students' explanations. So when we were on that method slide, I talked about how the participants described these three subsystems before talking about biofilms. And in that part of the interview, we saw that the majority of our participants did not know the functional definition of gene regulation. And in our normative model, we saw that these connection between cell-cell communication, the gene regulation to phenotype is a very important framework. And this gene regulation step is being skipped by our students. So the omission of gene regulation, and by extension that of proteins, prevents our students from fully describing the transition point. So then that led us to think about, okay, well, if the students are struggling to bring this in, why might that be? And so we theorized that the students' mechanistic reasoning within our biofilm context is possibly limited by transfer of knowledge. So when students are learning about various concepts and they're being asked to transfer those ideas elsewhere, that can be difficult if the change in context is what's called far. So is how similar, how related is this context or this idea related to other ideas? So even though we talked about gene regulation, cell-cell communication, and phenotype at the beginning of the interview, students then taking that information and applying it to the biofilm context may have been too difficult, may have been too far of a transfer to see what's going on. So our instructional implications then, we suggest scaffolding mechanistic reasoning into the classroom when you're exploring different processes in biology, and discussing how those integrate with other processes. As we saw in our data, there's definitely some growth that needs to be had in this area for describing mechanisms. And there are two models that are out there that you can use to help students practice mechanistic reasoning, called the MOC model or the MASH model. We also want to be aware of the situational nature of knowledge. So as I was talking about before, when students are learning about some sort of concept, it's generally within the context of something. So if they're in an ecology classroom, for example, they may be thinking about that knowledge only in the context of ecology. And when you ask them to use that same knowledge in a biochemistry classroom, that can be a really large leap. So consider the knowledge that is being taught and whether or not that knowledge can be easily transferred to different contexts. So that can be something to also integrate into the classroom, is practicing transfer of knowledge and helping to scaffold that transfer. We also suggest cueing prior knowledge and student reasoning. We had assumed earlier that by priming students earlier in the interview that then they would be able to talk about these things, but that transfer of knowledge was too far. And so to help students then to actually engage in the process, giving them some more cues at the time of assessments can be really beneficial. So asking them to explain their reasoning, to think about the ideas that they talked about earlier, can really help students put things together and answer these interesting biological questions. Thank you so much for listening to our talk, and I hope you get the chance to read our paper. I'm really proud of our work, and I'm hoping it's informative and can help future microbiology educators teach biofilm in their classrooms.
Excellent. Thanks. So this was a great example of qualitative disciplinary discipline-based education research, right? So we call it DBER, or biology education research. And I really like that you focused on systems biology, because it's one core concept in vision and change in the ASM curriculum guidelines, but there's just not that much research out there about how to do it. You know, there's these complex relationships, especially amongst scale. And I've always struggled with, how do we teach these complex relationships at different scales to our students? So I think that this is a really good model of how to do it. Can you tell me a little bit about the qualitative nature of this study? Like you said that you had 9 students that you worked with. Can you talk a little bit more about the length of interview and the selection of students for your interviews?
So the interviews were about an hour long. They lasted between like 20-30 minutes to an hour, just really depended on how long the student wanted to talk. Let me see. What was, I already forgot the other question.
And so how did you go about selecting your students?
Oh, right. So we wanted specifically biology major students to be a part of this study. And so we sent out an email to the biology majors listserv, and anyone who was a biology major was fit our selection criteria. We looked at their demographic. We asked them about what classes they took to see if any sort of, if, you know, being a senior where they've maybe had more biology classes, would those students potentially perform better than our sophomores who have had less classes? And we didn't see any relationship there. So we left it open to have any class standing could participate in the interview.
And following up, a participant asked, I'm curious about the characteristics of these students. So this is related. The student who had a more advanced understanding, do you have evidence to suggest that student was farther along in the biology curriculum?
So this is funny. That student was actually a sophomore. So it's likely that during the fall semester, they are taking their, Stephanie, is it the genetics, or which course are they taking in the fall?
In our curriculum, they take cell biology course in the fall semester of their second year, and genetics in the spring of their second year. So this student would have been in a cell biology course.
So they may have been able to use some of that information from their class to help them do better in the interview. That's entirely possible. But we had other sophomores who did not have that interconnected model. So just variance in student knowledge.
Excellent. And this participant said, really wonderful detailed work. Could you please share an example of differentiating between macromolecular versus molecular?
Yes. So let me pull up. We have our so if you go to the article and go to our supplementary materials, you can see all of our code books. So describing how we categorize various things and our definitions for them. So the main difference was macromolecular are going to be things that are at a larger scale than molecular. So for example, our molecular entities mostly included things like signals. Whereas like the macromolecular category was like mostly like proteins or genes. Let's see. So just larger structures within the cell. So, when we think about monomers being like the smaller pieces that are making up the bigger pieces. So we just had to differentiate in our code book about what we considered something to be more molecular, more smaller versus something that is built from the smaller pieces.
That helps. Okay. Let's see. Let's think a little bit more about the students. The participants want to know, were any of the 9 students currently doing work in an undergraduate research lab in an apprenticeship or in a mentorship model that was, say, outside of their regular coursework?
We've had so many students that some of their information starts to blur together. I know some of the students did have undergraduate research. I think there was maybe 1 or 2 in this study, but that did not seem to impact the answers that they gave. And maybe a related question, we also looked to see if any students were enrolled in a microbiology course, which would also, could lend some advantage to talking about biofilms. And we saw that there was 1 student who was currently enrolled, but that they hadn't talked about biofilms.
And just to add about, we had 9 students, right? So with 9 students in a qualitative study, we're not aiming to make generalizations to any, biology majors. It was just the features of student knowledge for these particular 9 students. So it's a good starting point that opens up some future directions and questions, but we weren't aiming, which are valuable, but they don't set us up to make generalizable claims. Just as a note about, it's 9 students, so we try to be very careful about what we're claiming, and we are richly describing what these 9 students did and said and how they thought, but we can't make any claims about how this relates to any biology major.
I think that's a really important point, because one reason we do these Jim B Live webinars is for our audience to kind of learn the differences between qualitative and quantitative research. And so thinking about an N equals 9 and what we can gain from that and what it does not mean. Let's see. Were your students able to draw? If no, do you believe there could have been an issue with displaying the transfer of knowledge due to having no way to describe the mechanism in words?
That's a really good question. So they did not do drawing in this study. We actually did a follow-up study where we did ask students to draw. And it does appear that being able to draw out how they think that these processes are connected may have an impact. That study was done, though, in a different context, so unsure, to what extent the answers would have been different in the biofilm context. But we do have some evidence to suggest that the students, if they are able to diagram, that kind of helps organize their thoughts, helps them explain it, and see those connections. But yeah, unfortunately, we did not do any drawing in this study, so only looking at their verbal explanations.
You think you may compare research performing universities and more class-based schools? So I think that this question goes to, this research was conducted at a big research university. Do you think that, could you state anything about what would happen if you did this at a smaller institution that didn't have so much high research going on?
Yeah, that's
Stephanie,
going.
do you want to take that?
Yeah, I mean, that's going back to our scope of inference, right? Because we were only, this is like, within a single institution, just a single institution, never mind one with a very high research activity, we can't we can only speculate. We would actually need to collect those data. And if we had a research question that was specifically aimed at trying to reveal any institution type specific ways in which students think, we'd have to do a really deep examination of it. That opens up there's a whole other research question to answer, and you would need to figure out how many students from different numbers of a similar institution type would you want to examine. What was it there? How was their curriculum structure? What were their learning experiences like? I mean, we'd have to gather a lot of really additional data to be able to characterize things well, to make those comparisons across students. And so, yeah, that long story short, it's interesting. It is important because we cannot make generalizable claims about any, other, students other than these 9 who are in this particular context. So being very careful with our small n study at a single institution. We can only describe those students and not even all students at this institution where they were recruited from. So there's trade-offs, right? We get really deep information from a very small number of students, but you can't generalize. So it could be that there are some really interesting and important differences.
This attendee is curious if you have any ideas or examples about how an instructor might scaffold the learning context between no context and biofilms. Does that make sense?
So talking about these ideas not in a context, and then how you would talk about it within a particular context?
I think so.
So I would say that, since knowledge does has the situational nature to it, I guess you would first need to figure out what is your goal in teaching it. So if your class, if you're looking to teach, we'll just say gene regulation. What is the purpose? What sort of context do you want the students to be able to talk about gene regulation in? And ideally, students should be able to talk about gene regulation in a variety of contexts. So my I guess suggestion would be to first lay out what is gene regulation, what are the steps, what is this process, and just describe the mechanism using the features of mechanistic reasoning. We saw in another study where we actually had instructors describe some of these processes that they also didn't always use mechanisms. So being aware that if you want your students to describe things as mechanisms, you should also be describing them in the form of a mechanism. And then after going through kind of generally like this is how gene regulation works, finding some interesting context to put it in to help the students like make the connection between, okay, this is what happens generally outside of a context, which may be kind of vague or ambiguous, and then situating those ideas in a context that's relevant for your class and embedding those different ideas.
And borrowing from what has been put forth by Harold Modell in physiology education, and I was trying to find a good citation. I can put it in the chat, and that related to what Sharleen was saying is being very explicit in cueing students about what he calls general models, right? So you have being very explicit, saying, okay, you know this about this process, okay, apply that model and identify how the little tiny differences that may exist given a different context, but it's the same general model. How is it behaving in this context? How is it behaving? And calling that out for students and being very explicit. So that I put a citation or a DOI in the chat and to share that, borrowing from an approach that's been championed in physiology education for general models that just keep coming up, like flux keeps coming up over and over and over again in physiology, for example. But it's that same idea, and being explicit to cue students to apply and transport that over and explore with them. How is it similar or different in this given context?
Yeah, the literature on knowledge transfer could also be really useful here. So looking to see, when you're having them apply these ideas in different contexts, what sort of contexts are kind of the most maybe flexible, or the most that students will be able to apply to, and before moving on to more difficult contexts or larger contexts.
Let's think a little bit about the curriculum and the implications for curriculum. In your opinion, do you think the scaffolding of the mechanistic reasoning for systems should occur within each course or across the major, or both and?
Well, my opinion is that it should be everywhere. I'm biased though in that thought. No, not okay.
No, I think the literature suggests, yeah, I mean, no, I feel that way too. I would say that biased, but I think that the literature supports that you need repeated opportunities for practice and scaffolded elaboration and across the curriculum, as Sharleen noted in describing that one of the students who had the most sophisticated model was someone who was a sophomore, right? But they're more proximal to maybe learning some of those, and that it's possible that maybe our curriculum doesn't reinforce and help the students to keep developing those ideas, right, across the curriculum. And I think the literature and education would support that you need repeated practice with feedback.
Participant asked, I wonder if students had any difficulties using appropriate words in describing the phenomenon. Did their term knowledge influence their conceptual knowledge? And how did you address those while analyzing the interview data?
Okay, that's a multi-part question, so I might need to hear some of it again. But so yes, we did find when we were examining, for instance, what entities they were using, that not all entities they chose to describe or include are correct or relevant. So for example, one student mentioned the nucleus. So bacteria cells don't have a nucleus. Or they talked about they would call things like stuff because they maybe didn't have necessarily a term for it. So when we were analyzing the data, we had our list of expected entities that we from the model and the description that we made. And we were cognizant of the fact that students aren't going to know very specific things like the names of this particular protein, or different operons, or all those different specific entities that actually go into that transition point. And so we wanted to make it more broad. So instead of describing, oh yes, the bacterial cells are producing this specific protein, that didn't matter to us. All they had to talk about was some sort of protein or some sort of gene. So we were very careful in that the entities that we were looking for, or that the way that they were describing it, would be reasonable for a student and not as very vetted out expert explanation.
And figure 2 kind of highlights that with the top box with some of the very specific entities, and then what we a transcription factor is the exact name. So if you want to look at what we how we did that and simplified things that's reasonable for the students, that's in figure 2 of the paper.
Thank you. So there were some students higher along or farther along in their undergraduate career that did not present making the greater connection. Okay, let me start again. Do you think that students farther in their undergraduate career did not present making the connection due to assuming that the knowledge is unspoken? If so, how do we help students not make assumptions and get that knowledge from their head?
So that might be the case. The interview methods that we were using was we asked them to think out loud. There was a lot of prompting. So for example, when we were looking at that biofilm figure, I asked them what do you see? What do you think is involved? What things that aren't that aren't visible in the diagram? What do you think the invisible things might be involved? And so we fully described all those things. And so we, in the question where we then asked them to describe that transition point, I pulled up those ideas that they had presented and said, okay, what about this thing you mentioned earlier as being important? How's that part of this transition point? And so it was a kind of a longer process of talking about that transition point and then asking them to then put it into some, like a series of events, which then we described. So the things that they talked about were supposed in, to the best of our ability, we asked them to think out loud, to speak that unspoken knowledge in a way. And so the protocol is limited and that we don't know the full scope of their knowledge, but to the best of our ability, we try to elicit that.
It is an important consideration, right? The degree of specification and yes, people don't always know exactly what level of detail you want. Sharleen, having read the transcripts, right, it did a lot of prompting to try to the best of her ability to really get at what they were thinking. And if they were pausing, what are you thinking about right now? And could you explain what you mean when you say X? But correct, you never get everything that's in there.
So here's where the method of the semi-structured interview really comes into play in being a skilled interviewer, right?
Absolutely.
Let's see. I realized that this was conducted in a higher ed. Do you think that the method design can be applied to K-12 students?
Absolutely.
Some really great studies. So we, cited one of them, that figure that was showing had the heart at the very beginning. It had the heart and had broken it down levels of organization. There have been some really great studies even on transfer in the context of like mechanisms that have been done more in the field of learning sciences, but Duncan and Reiser that they've done and colleagues have done a lot of that, and a lot of the mechanistic reasoning literature is actually in K-12 settings to begin with. That's been applied in higher ed.
And then if this person really wants to introduce biofilm reasoning into K-12, what do you think? Good strategy for K-12, something that all K-12 should have?
I think biofilms are really cool. And I remember when I was taking AP biology talking about biofilms, and there's what's really interesting. So I mentioned earlier that there are very few empirical studies that have been done in the context of biofilms. There's been at the time of the publication, there was only 3 studies that I found, and they were not studies per se, but descriptions of activities. So you can find those citations in the paper. But I remember like one of them was done with kindergartners, where they looked at they had these like gels and whatever, and then there was another activity I saw that was kind of more like public science, where it was like kind of like an event that a library or a department could put on, where they had like these containers that they put stuff on, and then you had to like spray it with a bottle, and you can see that, oh, if it's in the biofilm, the spray doesn't work, it doesn't destroy the biofilm, whereas if the bacteria by themselves, then you can like spray them off. So I think there could be really fun ways of including this in K through 12 that doesn't have to be specifically like, oh, we need to know all the little pieces of this mechanism. It can do a little both.
And it would really align with the next generation science standards and integrated STEM units, right? Creating you have to have knowledge of chemistry and engineering, and using biofilms in water treatment plants and digestion, right? There's all kinds of practical applications, and we have some integrated STEM K-12 education researchers here that I think that this would be perfect for one of their lesson plans across a variety of levels and line up really well with the next generation science standards and engineering practices that.
Thank you so much for talking with us today. That brings us to the end of our event. So we recorded this event and we'll put it onto our YouTube page, and you can find Jimby's YouTube playlist through our Jimby Live homepage. And we have another Jimby Live in 2 weeks. It's going to be really exciting. We're going to talk about looking at the microbiology concept inventory and how to use that to improve microbiology curriculum programs. So all y'all microbiologists, please come back in 2 weeks. And again, that event is free. So with that, we'll close this out. Thank you so much, speakers, and we hope to see you in another event.
Thank you so much for having us. And those are fantastic questions. I really enjoyed this.
Yeah, thanks,
Yeah,
everyone.
great audience all the time at Jimby Live.